Nowadays, plant research and conservation widely uses plant tissue culture method to further the growth of some species. Intensive sterilization is a mandatory procedure to eradicate any foreign microbes and to further improve optimum yield. Traditionally, reliance on chemicals was used widely for its effectiveness. However, chemical sterilization has its negative traits such as necrosis and the difficulty in disposing those used chemicals. This particular research uses a compelling new method that could be used for sterilizing explants for in vitro culture, which uses an ultrasonic sterilization method. Contrast to the conventional method, ultrasound alternatives is considered safe for environment and are cost-effective. This project focuses on selected Solanaceae species, including various capsicum species and tomato, using seeds as the explant source for in vitro culture.
Main challenge in plant tissue culture is that conventional seed sterilization relies heavily on hazardous chemicals. Although these chemicals are effective in removing contaminants, they pose safety risks to users, generate chemical waste, and raise environmental concerns, especially in teaching and laboratory settings. Therefore, the objective of this study is to develop and evaluate an ultrasound-assisted teaching aid as an alternative sterilization method. This approach aims to provide a safer, greener, and more sustainable technique for plant tissue culture education by reducing the use of chemical sterilants while maintaining effective seed sterilization.
Now, I will explain the methodology used in this project. The workflow consisted of 5 main steps: sample preparation, ultrasound treatment, aseptic inoculation, culture observation, and evaluation. First, seed from selected Solanaceae species, including capsicum species and tomato, we are used as the explant source. The seed we are prepared and handled individually before undergoing ultrasound treatment. Next, ultrasound frequency of 33 kHz and 40 kHz we are applied as the main sterilization treatment to reduce the dependency on conventional chemical-based sterilization methods. After the treatment, the seed we are transferred using aseptic techniques and inoculate into MS media for in vitro culture. Finally, the culture we are observed for contamination, necrosis, and seed response to evaluate the effectiveness of the ultrasound treatment. Overall, this methodology demonstrates a greener, safer, and more sustainable approach for plant tissue culture learning. That's all from me. Thank you.
To conclude, green culture demonstrates that ultrasound-assisted sterilization is a practical and sustainable alternative to conventional chemical sterilization in plant tissue culture. By reducing the use of hazardous chemicals, it creates a safer laboratory environment while promoting green laboratory practice. At the same time, it provides undergraduate students with valuable hands-on experience in aseptic techniques, contamination control, and plant tissue culture. For future development, we recommend evaluating additional ultrasound frequencies and treatment durations, as well as testing the method on a wider range of plant species. Integrating digital learning resources such as QR codes or instructional videos could also enhance students' learning experience. Overall, green culture is an innovative, safe, and environmentally friendly teaching aid that supports sustainable plant tissue culture education.